Hi everyone, my name is Sarah Downham and I'm going to be giving my final presentation on congenital adrenal hyperplasia. So first I'm going to start off with just giving a general overview of what congenital adrenal hyperplasia or CAH is. So congenital adrenal hyperplasia refers to a group of genetic disorders that affect the adrenal glands. And the adrenal glands produce important hormones like cortisol, shown here, which regulates the body's responses to illness or stress. And CAH is an inherited disorder that results in low levels of cortisol and high levels of male hormones, causing the development of male characteristics in females, and also early puberty in both boys and girls. CAH affects about 1 in 10,000 to 1 in 15,000 people in the United States and Europe, and in select populations such as the Yupik of Alaska, the prevalence can actually be as high as 1 in 400. So I was just going to give a brief history of congenital adrenal hyperplasia since it's been around for quite some time. So it was discovered by Luigi Di Crecchio, an Italian an anatomist in 1865, and he first documented CAH in a female patient with enlarged adrenal glands, male-appearing genitals but no testes, and an internal female reproductive system. And so fast forward to 1962, where a genetic link to CAH was discovered, and this genetic link was found as 21 hydroxylase deficiency, where each parent passes one copy of an abnormal gene down, which results in the child having a double dose of the abnormal gene. And I'll talk more about that later. Then in 1977, scientists discovered a method for detecting 21-hydroxylase deficiency in newborns based on the level of hormone of 17-hydroxyprogesterone. And then in 1978, the specific location on the gene that causes CAH is found, and I'll also talk about that a little bit later. And then in 1984, a female fetus affected by CAH is treated prenatally with the administration of dexamethasone to the mother. And then lastly, in 1989, the United States began to include CAH on their newborn screening programs. So next I'm going to move on to the symptoms of CAH. And so people with CAH often have enlarged adrenal glands, and these adrenal glands produce a large amount of androgens. And this leads to atypical development in female sex organs. And oftentimes females will have ambiguous external genitalia, even though they are genetically female. They will also not have any normal internal reproductive organs, and males will not have ambiguous genitalia, but both males and females may go through puberty earlier, have fast body growth, and have short stature. And 75% of people diagnosed with CAH also have an aldosterone deficiency. And this deficiency leads to the inability to retain salt and water, which results in excessive dehydration, hypovolemia, and hypotension. And this can lead to profound weakness, vomiting, diarrhea, and circulatory collapse. But luckily, the vast majority of children are treated early, so they do not have to experience these complications. So now that we've talked a little bit about the symptoms and the background of congenital adrenal hyperplasia, I wanted to spend a couple of minutes going into the genetic causes of the disorder. So there are a few different types of CAH, but the most common is due to a deficiency in 21-hydroxylase. And deletions and mutations in the CYP21A2 gene lead to CAH due to this 21-hydroxylase deficiency. And so the full name is cytochrome P450, family 21, subfamily A, and polypeptide 2. And so 21-hydroxylase plays a role in producing hormones cortisol and aldosterone, and these hormones are responsible for different functions. cortisol is the body's stress hormone, so it protects the body from stress while also decreasing inflammation. And aldosterone regulates the levels of salt the kidneys reabsorb. And these levels of aldosterone can increase or decrease blood pressure. And so here we see a normal adrenal gland with normal amount of cortisol, aldosterone, and androgen hormones. But the adrenal glands in CAH, like I said, lead to that decreased cortisol and aldosterone and increased androgens. Excuse me. So we're just going to talk a little bit more about the genetic causes of CAH. And so the majority of mutations, about 70%, are from gene conversions, which is the movement of mutations to the active CYP21A2 gene, whereas the other 25 to 30% are from large deletions. And we can see this gene conversion in this picture here. And CAH is inherited in an autosomal recessive fashion. And so mutations in the CYP21A2 gene cause a deficiency in 21-hydroxylase, which leads to CAH. And these deficiencies lead to a buildup in the adrenal glands, which are then converted to androgens. And as a result, there are abnormalities in sexual development. Further, 21-hydroxylase is a cytochrome P450 enzyme, which are involved in different processes in the body. And for example, they help with reactions that break down drugs and produce different hormones. So now I'm going to move on to talk about the mode of inheritance of CAH. And so like it says here, CAH is inherited in an autosomal recessive fashion, meaning that an individual inherits an abnormal gene from both parents. So if the person has one abnormal and one normal copy of the gene, then they will not be affected. They will just be a carrier for the disease. So that's shown by these two children here. But because CAH is inherited in an autosomal recessive manner, in order for the offspring to have CAH, they will need to be homozygous recessive for the allele. And this child here will have the condition because they inherited both non-working genes. So therefore, the risk for two carriers to pass the abnormal gene down to their child is 25%. And the risk for the child to be a carrier is 50%, whereas the risk that the child receives both normal copies is 25%. And that is shown by this child here. So now I'm going to just move on to describe a little bit more of a detailed pedigree. It might get a little bit cut off by my picture, but I'll try to move it around as much as I can. And so like I said, it's inherited in an autosomal recessive fashion. And so the blue square as shown up here in the right-hand corner and right here as well indicates that that person has CAH. And so, for example, for Michael and Julie, there is a 50% chance that their children will have the disease and a 50% chance that their children will not have the disease but will be carriers. For Patricia and Matthew, shown here, there is also a 50% chance that their children will have the disease and a 50% chance that their children will not have the disease but will be carriers. For Harrison and Olivia, shown over here, they are both carriers of CAH, so the risk for two carriers to pass down the abnormal gene to their child is 25%. The risk for their child to be a carrier is 50%, and the risk that their child receives both normal copies is 25%. For Joseph and Mia, shown here, there is a 50% chance that their children will have the disease and a 50% chance that their children will not have the disease but will be carriers. And then for James and Ava, shown here, there is a 100% chance that the offspring will have the genotype with a phenotype of being affected by CAH. So there are a lot of different options for genetic testing for CAH, and they can be done prenatally or after birth. And so there are, like I said, there are several different genetic testing options available. And first, they can be diagnosed prenatally, either through amniocentesis or chorionic villus sampling, as shown here is amniocentesis. And then over here is chorionic villus sampling. And genetic testing for CAH via an amniocentesis can be done between 15 to 20 weeks, and testing for CAH via a CVS can be done between 10 to 12 weeks. And testing for CAH after birth via newborn screening panel is ideally performed 24 to 48 hours after the baby is born. And after birth, the newborn screening panels can also diagnose CAH. And results can vary from normal, borderline risk, and high risk. And all of these genetic tests look for mutations that lead to deficiencies in the gene 21-hydroxylase. Testing for CAH via amniocentesis or CVS is completely optional, but the newborn screening panel, however, is mandatory because if left untreated, the baby could develop a lot of serious problems. So next I'm just going to talk a little bit about some of the controversies surrounding these genetic testings. So there are controversies surrounding newborn screening as well as prenatal screening via amniocentesis or CVS. And in regards to newborn screening, the controversies mainly surround obtaining parental consent for using residual blood from the testing. And in terms of amniocentesis and CVS, the controversies lie surrounding testing for the diseases prenatally. So some people believe that testing for genetic disorders prenatally can lead to a rise in abortion rates. And further, there is a small rate of miscarriage. And it could also lead to sex-selective abortions. And parents may also become more anxious while waiting for these results, which can have negative effects on the pregnancy itself. However, with all of this being said, early detection of CAH is important because if treated soon after birth, these children can go on to become healthy members of society and have healthy growth and development. And it's also important because a diagnosis of CAH early on can prevent death because without early treatment, many CAH patients can lose too much salt and pass away very early in life. So thank you so much for listening. I hope that you enjoyed this presentation, and please feel free to reach out to me if you have any questions at all. And this is my first reference slide, and this is my last reference slide. Thank you so much.